Okay, do you hear me? Did you hear me?
Yes, master.
Okay, now class begins. Okay, at the first of this class, I have something to inform to you, because the time point of tomorrow afternoon is unavailable because we have another class about the teacher Liu Jie, right? So the time we have no available time for this class. So the time of this class on tomorrow afternoon will change or return to the original time. That means tomorrow we have no class, and on Wednesday evening we have the class. The monitor was informed to you, right? Okay. So afternoon, the evening of Wednesday, we will have a class. I will at that time before the class, I will inform you in the Dindin and the number of the classroom. Yes. So this class we will introduce something about the complement system. The complement system is a very important system for the immune response. In the last class lesson, we will talk about we will mention the function of antibody. After the antibody, sorry. After the antibody binding to the epitope of the antigen, the antibody will have a conformational change and will show out the CH2 domain. This domain will bind to a protein. It means CQ1 or C1Q. This protein will as an initiator to activate the complement system. This pathway is one of the complement systems. So today we will talk about more detail about the complement systems. At first, we will see the definition of the complement system. The complement system is a group of plasma and cell membrane proteins, most of which circulate as inactive enzymes and are activated at the site of inflammation. Pay attention here. Is two properties here is most of them is in the plasma or cell membrane and sorry. How to
Zynogens.
engines. The components of complement system before activation is inactive. Zymogens, and after the active, they will perform the function here. And the components of complement system, some protein belong to the complement innate components, such as C1 to C8, C9, and B factor, and another something about C1INH, I factor, H factor, and another is belong to complement regulatory proteins regulation. It is regulate the process, and another is the CR. CR is the receptor molecule present on different cell membrane that bind to active fragments formed during complement activation and mediate a variety of biological effects, including CR1 to CR3 or CRAR or C5 RAR. R here means receptor. And the nomenclature of complement system. The classical pathway and terminal components of complement are named in order to discovery, in the order of discovery. And the components of alternative pathway are represented by capsulator, and the components molecular which with enzyme activity are marked by horizontal lines above them. So here is a line above the C4B2A. And this component is a complex and with a C3 digestion activity. And a fragment of complement cleaved during activation, each of which is named after the suffix of the complement components in lower case. Here show the C3A and C3B. Both of them came from the C3. After the digestion, the C3 were divided into or cleaved into two parts, two fragments. The one bigger fragment named C3B, and the small fragment named C3A. And the sum of about infection, acute inflammation, and tissue damage state. The components of complement is vulnerable to a variety of physical and chemical factors. Well, the components of the complement system will lose the activation at the 56 degree for 30 minutes. After if you use the if you put the complement system and components in under degree of 56 for half an hour, these components will lose the function. And 5, the pathway of complement activation. Generally, we will talk about 3 kinds of pathway here. Remember, the components of the complement system have no function before activation, but the first member of this pathway will active, and the follow step one by one will continually to happen. So this process named enzymatic cascade, just like a domino cat. The picture show here. If you put so many domino and a layer to or another kind of shape, a little piece of domino give power to the system. The next step will all the domino will touch it one by one, and the end is a cascade process. So this process is the key point here is the initiator. The three pathway here: classical pathway, alternative pathway, and MBL pathway. At the beginning of the initiation is different each other, but the following the rest of those 3 processes will share with the same process. So even we talk about 3 kinds of pathway, the finally results are the same, and the difference between each other only on the beginning of the initiator. So we introduce it one by one. The classical pathway, the initiator is AG bind to AB. Here AG is antigen and AB is the antibody. The antibody generally is IgG1 or IgG2, IgG3, IgG1 and the IgM. The IgG1 or 2 IgG1, IgG2, IgG3 is the subgroup of the IgG. After the antibody binding to the antigen to become complex. It means immune complex. And the next step, the C4, C2, C3 will attach it and take part in the process. We can see here the components of this pathway, the C1 on the first site. So we can see the first protein or compound. Sorry. Oh, no complex, right? Complex. Oh, sorry. It's right. It's right here. So the initiator is antigen, the binding between antigen and antibody. It become formed IC, and this IC will recognized by the C1 protein. Here we can see the C1 includes three part subgroup, three components. We can see here the C1q like a bunch of flower and placed upside down, and in the middle of the C1q there are two C1R or C1S and three kind of protein together to make up a complex to perform the function. And on the other hand, the antibody binding to the antigen will have obvious. Here is the IgM binding to antigen will have a great change in the shape. So this change on the conformational, the conformational change will bind into the C1 complex here. We can see on the below the IgG bind to the green point. Current cycle means the epitope, and the IgG binding to the epitope, and the C1 binding to the IgG, and the below the IgM bind to the epitope, and the C1 also binding to the IgM. So this this IC binding to the C1 is the very key step for initiation of classical pathway. And the activation requestion is the formation of Ig complex, and only IgM or Ig3, Ig31, Ig32, Ig33, IgG3, sorry, IgG1, IgG2, IgG3, and more than 2 complement by sites exposed. Here is the step to introduce the detail. The antibody binding to the antigen on the surface of the antigen, and the C1 will bind to the antibody, and the legs, the C1s have the activity to digest the C4. So the C4 will divide it into big fragments, small fragments. The big fragment is named C4. Is named C4A. However, after the produce of C4B, create, and then the C2 also digested by the C1s, which the C2 divided into 2 parts. The small one is C2A, and the big one is C2B. Here, the C4B and the C4B and the C2A together to form the complex. Here is named C4B2A as a layer belong to them. This complex have ability to digest the C3. Together to form the new substance. There is no enzyme activity here to digest the C3. After the formation of C4B and C4B2A formed, and the next step is C3 recruit, because and then this activity will focus on the C3, and the C3 will digested into also 2 pieces of here, two fragment here. The one is C3B, and the small one here. The complex will add a new member. It's named C3B. The C4B, 2A, 3B together is a new complex here, and this new complex have a new amazing activity, enzyme activity. The activity is to digest the C5. So these components, this complex named C5 convertases, C5 converters, because this complex will digest the C5. So reasonably, we can see the C5 will digest the C5 into two fragments, C5A or C5B. So this process is clearly, I think, because the immune complex formed and recognized by the C1, and then recruit the C4 and digest the C4 into C4A is run away, and C4B lived. C4B recruit the C2 and furtherly digested the C2 into. Sorry, it is wrong. I have some wrong here. No, is C2, CP2. Not. No, it's right. C4B and C2 together, and the C2 will be digested into two fragments, and the 2A remained. So the C4B 2A together, we call it C3 convertase. And the 3 convertase digest the C3 into together with formed a new complex. It named C4 convertase, and the C5 convertase will digest the C5 into C5A and C5B or C5B, right? Here, the very key step of this pathway is the conformation, the formation of the 3 convertase and C5 convertase, because these 2 convertase were the key enzymes for the cascade process. The second pathway is the alternative pathway. The initiator is not the IC, is not the immune complex, but is the bacteria or some LPS or peptide glycan or other substance on the surface of the bacteria. And the components of this pathway are other factors, such as factor B, factor D, or the factor P. Here we can see this pathway. We can imagine that the C3 is the same components in the classical pathway. So It's just what I see. In the beginning of this three pathway is different, but the end of the end of those three pathway will share the same progress, the same process. The end of those pathway are the same from beginning of the C3. So we can see the diagram about the alternative pathway. The C3 in our body liquid is spontaneous and lost very quickly, and lost, he will produce and lost very quickly. But after the after bacteria or something or virus come here, the C3b will bind to the bacteria or virus, and then B factor and D factor bind to the C3b, and this structure named C3 convertase. So this pathway, the three convertase is different from from that of classical pathway. And the C3b and BB, these components will have ability to digest the C5, and the next, the C5 will, the C5 convertase will formed. Arrived at this step, the 5 convertase formed. The next step is the same to the classical is the same to that of. So we should be attention here. In the alternative pathway, and the C3 convertase is C3b and B, capital B and the capital B together, and the C5 convertase is C3b and BB and C3b. Two 3b here together. Also the three components on the C5 convertase. It is the obvious difference between the alternative pathway and classical pathway. So here is also model pathway here. The C3 spontaneous conversion or produced by classical pathway, and then got to the bacteria, and B factor and D factor recruit and to form the C3 convertase, and the 3 convertase will furtherly digest the more 3 and the C3 much more C3 produce, which give a positive feedback loop. And finally, the C5 convertase formed, and the next step will one by one to active. So in this way, in this pathway, the form of 3 convertase and C5 convertase is the more key, is the key point or the key step for the activation of this pathway. And the third one is the MBL pathway. And the initiator is very different between the alternative pathway or classical pathway. This pathway is the initiator of this pathway is contain carbohydrates. It means mannose expressed on the surface of the microorganism. The MBL is the mannose binding lectin. It also including the MPS. The MASP is a MBL associated serine protease, and the components of the MBL pathway also including C4, C3, or C2. Actually, the pathway, the MSP is on the structure is similar to the C1. So we can imagine that this MASP here is the replaced substance of C1 activity. So the C1, C2, C3 is the same components. The C1 digests the C4, and here MASP also digests the C4. After the formula of C4b produced, the C2 will recruit here and take part in. So here is the single pathway of the step shown here. The MBL binding to the mannose, and the mannose, this binding is active the MASP1 or 2. The MASP1 or MASP, here is MASP2, will have 2 kinds of enzymes activity. The one is digested C4 to C4b and C4a, and another activity of this protein is digested the C2 to C2a, C2a or C2b. Similar to the classical pathway, the C4b and C2a together to form a complex. Here is the same structure or the same constant with the classical pathway. So here also named the C3 convertase, and furtherly the 3 convertase will digest the C3 and get together with to form the 3 components factor. The structure named is C5 convertase. And we also here we already know the single pathway of 3 kind of activity pathway, and we also knew that the end here, the key step of here, the step of this pathway is the formation of C5 convertase. Each pathway, the end of each pathway, on the each pathway of the this The key point or step is the formation of C5 convertase, and the same activity that is to digest the C5 to digest the C5. So the terminal sequence is C5 to C. And finally, the membrane attack complex will produce product. You can see here, after the C5 convertase formed, and the C5 will digest it into C5A or C5-2 or C5B, and the C5B will together here and insert into a membrane and the cell membrane, and then recruit the C6 or C7 and C8 together, and this structure will finally rescue the about 15 C9 and to form a polyC9 to form a bottle or is I think I check the barrel. Sorry, so many about 15 about 15 C9 recruit by C5 to 8. It formed a big barrel and across the target cell membrane like this picture showed. The C9, so many C9 and formed a barrel across the target cell membrane, and we can imagine that the this structure named MAC, the membrane attack complex. The MAC result the outflow of the cell components, like this picture shows. The components of the cell contents will flow out because the barrel is very big, and so many barrels formed on the surface or on the cell membrane. The blue picture is showed a bacteria killed or digested by the MAC. The A showed original bacteria, and the B is showed formed in the process of the formation of system as a complement system, and the C present the result of MSC formed on the cell membrane, and the bacteria totally digestion, and here is cleaved. So the complement system, at the final step of the complement system, is formed an MAC on the target cell membrane. This step will lead to the cell components to flow out. And as a result, the target cell will cleaved by the complement system. So I think it's very simple logic problem. Here is total view of the of three pathway, and the three pathway also although the three pathway have a different beginning, but they have the same ending. That is to form the MAC on the surface of the on the membrane, cell membrane. This MAC is very strong power to digest to cleave the target cell. The target cell including bacteria and including tumor cell or including the cell contain the virus. And the comparison of the three complement activation pathway. This is a detailed comparison here in the table. And the second part is the biological function of complement activity. And the first is the opsonization. Opsonization will motion in the function of the antibody because after the antibody bind to the antigen, the Fc fragment of the antibody will recognized by the macrophage. This is help the macrophage to uptake the protein. So this process called opsonization. Here we also call the opsonization because the C3 also can bind to the surface of the bacteria. Is similar to the antibody binding to the surface of the antigen. And on the other hand, the phagocyte or macrophage also have the receptor here named CR1 can recognize the C3b or C4b. Also, this process can facilitate can help the phagocytes to get the bacteria easily or conveniently. Here showed the process here. C3b binding to the surface, and on the one hand, it will activate the alternative pathway, and on the other hand, the CR1 on the local on the phagocyte, and CR1 binding to the C3b, which can help the cytos to uptake the bacteria. So this pathway named opsonization.
Blanche?
What?
对，老师，I
I see.
think I cannot see your PPT，老师。
哦，有共享吗？哦，how about now?
It's okay now，老师，Thank you。
Okay, I see it repeated. Okay, the optimization is here is because the C3B, this here is the green color is the green color is the bacteria. Here in Chinese is 细菌. The bacteria on one hand is recognized by IgG, and on the C3B also can recognize the surface structure of the bacteria. And the macrophage also have the receptor, like just like here is CR1. The CR1 is have the ability to have the ability is to binding to the C3B. This binding will help the macrophage to uptake the bacteria easily. This process named opsonization. And the other function of the complement system is immune enhance. Immune enhance means the C3B coated immune complex are unable to bind to the CL1 of erythrocytes, which carry the immune complex to the liver and the spleen, where the ingested by the macrophages. You can say here the so many soluble. Sorry, I check the 可溶的 soluble. Sorry, soluble. The soluble antigen here will also bind with the antibody, and this soluble IC is not easy to clean, but here we can see the C3B bind to the FC fragment on the antibody. So, and the red blood cell have the CR1 receptor, and the CR1 will bind to the C3B, and the red blood cell and on the blood and arrive the liver cell, liver or spleen. In the liver or and the spleen, so many macrophage or another cytos to treatment those IC. Here is not just like it means immune adherence. Here is the key role. I think is the CR1 on the red blood cell. The red blood cell. The red blood cell just like a track transferred the IC to the spleen or liver, and the IC is fixed on the red blood cell by C1b and C1i, CR1. And then finally, the IC arrived the spleen or liver and uptake by the macrophages or phagocytes. And the third key function of complement system is inflammation, because the little fragment of C3 and C5 here named is C3A and C4 or C5A. This little piece, these two little fragment also have a very important function for our body. Deregulation of most cell and both cells by binding to C3AR and C5AR. This is two kinds of receptor on the basophil. Of those cells, which consequent release of so many vasoactive substance that induce inflammation. And on the other hand, C5A is strongly chemotactic for neutrophils, inducing the migration towards the site of the inflammation and increase the vascular permeability and cause vasodilation. So when we focus on the attention to the three active pathway, that we should be also pay some attention to the production of this pathway, such as the 3A or 3-5A. Maybe the C3A or C5A not the components for formation of MIC, but the soluble fragment as a soluble fragment, the C3A and the C5A also played a very key point in some physical process. Here we can see the inflammation is the development and I think is the initiation and the development of the inflammation displayed a very key relationship with the C3A and the C5A. And the C3A and the C5A is how to say, I checked the answer, is check the yes, is a byproduct. The byproducts, the C3A and the C5A are the byproducts of the activation of three kinds of pathway, complement system pathway. So it also, the function over here is also belong to the complement system. Okay, in this class, we should be know something about the activation process of three kind of pathway and the in some degree about the function of the activation of the complement systems. Right? So this class is finished. Okay, any question?
没有问题了，Chef。
Okay, okay,
Thank
thank
you,
you.
sir.
Okay, okay, goodbye.
Bye bye.
Okay,
Bye,
Alicia.
Alicia.
Goodbye.